Hi dear students, welcome to one more session of molecular biology. Today we are going to talk about the methods to detect recombinants. In the previous session, we had come across that once a recombinant DNA is being formed, they have to be transferred to a host cell, and we need to identify those host cells which contain the recombinant DNA. For that, we use the screening techniques. And in the previous session, we had mentioned two different types of techniques. One is the blue-white screening, when you use the LAC-based vectors, and the previous one, which we had discussed, was the antibiotic-based screening of the recombinant ones. Now, in this session, we are going to talk about a little more advanced technique called the Carbonyl hybridization technique. Now, what is a Carbonyl hybridization technique and when do we use it? A Carbonyl hybridization technique is used mainly as an advanced level screening technique to check whether the whole sequence of the gene uh, is being transferred towards the host cell, or if just segments are being there. So, though now if you have a small segment of the desired gene was only cut and gone also, you might get a recombinant in blue-white screening or antibiotic resistance and all. But to confirm whether the whole sequence of or a particular sequence of the entire gene is being present, you use a technique called the Carbonyl hybridization technique. Here, you are using the DNA sequence of the desired gene of interest, and you are taking each and every colony and checking whether that particular sequence is there in the host. So it's like a confirmation that your host cell does contain the gene of interest. At the nucleotide level, you confirm it. Now, how do you do this? So, uh, it's usually if you have an agar plate with the different colonies, the DNA as well as the RNA can be released from these microbial colonies. And this DNA and the RNA, which has been released from the microbial colonies, now how it is released? Of course, those cells will be denatured, and all those cells will be first blotted onto a particular membrane. Okay, or they are blotted in a sense they are being transferred onto a particular membrane, and the cells are being lysed so that the nucleic acids will be released from those colonies. And these nucleic acids which have been released from the bacterial colonies or the transformant colonies, they will be denatured and fixed to a filter and hybridized with a labeled probe. Now, what is a labeled probe over here? The labeled probe will be specific towards it is a single strand of DNA which will be complementary to your gene of interest, or it will mainly code which gene, or which is it will have a sequence similar to that of your gene of interest. It doesn't mean that the whole gene should be present, but a small segment of the gene of interest will be present. So if the colonies which you have obtained have that particular sequence, this probe will go and bind onto the those regions of the DNA. And usually before the probe is being added, you take the membrane and you also block it with some blocking reagent so that unspecific binding is being prevented, and excess probe is being washed away, and the membrane is then visualized under UV or autoradiography so that wherever the probe goes and binds, we can identify that those colonies will be having the gene of interest. So, Carbonyl blot hybridization is used for screening clones or even bacterial isolates. You can just have a look at the picture, then it will be more clear to you. Now, if this is a particular recombinant bacterial colony containing plate, what happens is you take a nitrocellulose or nylon membrane and place the membrane on top of the agar containing separated bacterial colonies. And there will be a master plate over here. So the master plate, all the cells are allowed to grow over here. You have it over here. And at the same time, you take the filter paper and it has been placed on a new agar. So invert the membrane and lay over the new agar surface and allow the colonies to regenerate on top of the filter. Now, after it has been allowed to grow, what happens? This filter paper is being taken. You take this membrane, you take this membrane, not filter paper, the nitrocellulose or the nylon membrane is being taken, and remove the membrane with attached colonies and soak them in successive solutions like denaturation, neutralization, washing, as well as dry and fixed DNA. During this process, what happens? The DNA of all these colonies, the DNA as well as RNA, will get denatured and they get bound onto this particular membrane. So now the membrane will be containing the nucleic acids corresponding to different colonies. And the bacterial DNA is now fixed onto the particular membrane. And after that, this membrane is taken and washed with a labeled probe. A probe is a segment of a single-stranded DNA, and it might be either radioactive or sometimes it might be fluorescently labeled. So you attached onto it, and now in this case, since we are using an X-ray film, it is radioactive. So consider that case right now. So you hybridize, so you take this membrane and onto this membrane, you will put the probe. So the if one particular colony does contain the specific sequence corresponding to the probe will come and bind onto it. And you wash the excess probe and then you go for developing it onto an X-ray film. So only those colonies, only those regions where the probe is being bound, you will get a blackening. So you can identify corresponding to this in the master plate which colony has that particular sequence. Now, I had told you about a blocking agent which was being added. See, sometimes non-specific binding before this probe is being attached here, non-specific binding on the background where the colonies are not there, that also could happen due to some similarity or any errors. So what you do is before the probe is being added, you will add a blocking agent so that all those regions where the DNA is not being present, the blocking agent will go and bind, and background based hindrances or background based errors would not be happen. So the probe will go and only bind onto those regions where the DNA is being present. So that's what about in this previous session we had talked about the blocking reagent. So before adding the probe, you will add also the blocking reagent so that the unnecessary or the unwanted binding at the background region will be prevented. And you take this X-ray film and you view it correspondingly, which is the particular colony. Or so you can identify this is the one which is having this colony is the one which is having the desired gene of interest, and then you can take this particular colony, grow it in a liquid culture, and take different colonies of that recombinant bacterial cell. So that's about the colony blotting which we have. And same technique, instead of using X-ray films, you can use other fluorescent labeled probes also can be used. I'm not going to the details of that because I guess this will be sufficient for you. Either you can use radioactively labeled probes or you can use fluorescently labeled probes. Now, the same principle of colony hybridization can be used in the case of plaque hybridization also. The only difference is that the plaque hybridization is used in a technique where you are using viral vectors. That is, if you're using, instead of having in this case now, look at this case, in this you are having bacterial clones and you are checking whether each clone is having your desired gene of interest. Now, in the case of plaque hybridization, you know what is a plaque? A plaque is mainly being formed when a virus affects, or now in the case of bacteriophage, a bacteriophage can grow on bacterial cells. Okay, and where all the what happens? The bacteria is being eaten up by the bacteriophage, plaques will be formed. So when you're using some viral vectors, sometimes after the process of transduction, plaques will be formed on the particular on the agar plate. To identify those plaques which have your gene of interest, you can use the plaque hybridization similar to the Carbonyl hybridization. Just a small overview is being given here. Here a filter paper or a membrane is being added onto it, and so DNA is being transferred onto this filter corresponding to the plaques, and then you will block the filter with non-specific DNA or protein, and you will hybridize it with a labeled probe, and then you detect it by autoradiography. So the technique of plaque hybridization, here you are using a plaque based colony, but the same procedure or the same principle has been followed for colony hybridization, which uses bacterial colonies. And here the bacterial colony based DNA is being transferred onto the nylon or the nitrocellulose membrane. It depends upon what you have. Okay, and in this case, what happens? The DNA or the RNA of the plaques is being transferred onto the nitrocellulose membrane. What is a plaque? You know, a virus affected bacterial cell is called a plaque. So in both the cases, you're looking specifically whether the gene of interest has is being present in your colony or in your plaque. I hope you are now clear with the plaque hybridization and the colony hybridization which you have here. And the basic procedure is just the same, and you check whether in these colonies you have your gene of interest by transferring the DNA or the RNA of those colonies as well as plaques onto a nitrocellulose membrane, and then you block them with certain blocking agents so that the non-specific binding will be prevented, and then you hybridize them to a labeled probe, either radioactive or fluorescently labeled, and then you detect them by autoradiography. So I guess you are clear with it. Thank you for now. I would also like to add a small note on the general principles of blotting techniques or the hybridization techniques which we are using here. So once lots of DNA is being transferred onto the particular membrane, you have to check whether your target DNA is being there. For that, you will take another probe DNA which would be able to bind to your gene of interest. Okay, or now this is a membrane here. Now the gel which is being used is for another process in which the DNA from a gel is transferred onto a membrane. But in our case, it is a colony from which we are, or a plate from which we are transferring the DNA onto the particular membrane. And the probe DNA, which is also a single stranded DNA, which would bind specifically to the desired gene of interest, will bind onto it, and you wash off the excess probe DNA, and then you apply it on an X-ray film, and you develop the film, and wherever the desired gene of interest is being present, you will find darkening over here. Now, this example which we have shown here is a hybridization where the DNA from agarose gel electrophoresis is transferred onto the membrane. But previously we were talking about what? We were talking about DNA getting transferred from the colony as well as from the plaque onto the membrane. But in either of these three cases, the principle is just the same, that a probes bind to a target DNA. And once it has been bound, what happens? You either detect it using a radioactive detector system or a non-radioactive detector system, and you check whether your gene of interest is present over there. Let it be from a colony, from a plaque, or if it is also on a agarose gel. So thank you for now.